Right then, so this is oxidative phosphorylation. Okay, so I'm going to draw my diagram on the right. I'm going to draw it on a bit of an angle so that it's clear that the energy is kind of flowing down when I do my electron transport chain. Let's start with a slanty membrane. There's going to be a couple of things on the internal membrane, so let's put them in first. Okay, so I've drawn my internal membrane on a bit of a wobble. That's because it's the folded internal membrane of the mitochondria. So let's label whereabouts in the mitochondrion we are. So this is the outer membrane. This one is the inner membrane. The gap between them has a name, and that's very creatively called the intermembrane space. And then the other side of the inner membrane must be, you should know this by now, the matrix of the mitochondria. Okay, so where are we going to begin? Well, we start with in aerobic respiration, glycolysis, link reaction, and Krebs cycle, and these are all generating some reduced coenzymes. So, for example, we have reduced NAD, which is written as NADH, and we have reduced FAD, which is written as FADH. These guys are reduced because they've gained some hydrogen and electrons, and they are oxidized back into their oxidized form. So, NAD and FAD. Now, this hydrogen atom is made of two things, and they're actually going to split apart here. So we're also going to get a proton here. The other thing, a hydrogen atom is a proton and an electron. So we also get an electron, which enters the membrane and goes into an electron transport chain, which last time I drew as a series of these electron carriers in the membrane. And the electron is going to flow effectively downhill, as I said before. That's why it's on a slant, through the electron transport chain, releasing energy. Now that energy is going to be used to pump the protons into the intermembrane space. And that creates our proton gradient, which, if you watch the lesson on chemiosmosis or the chemiosmotic theory, is how basically all the ATP, more or less, that we use is generated using this cycle here, which you'll be pretty familiar with by now. So it's all about creating this proton gradient and having the protons diffuse down their electrochemical gradient through ATP synthase. And this process, the blue arrow process, is the process of chemiosmosis. Okay, so more or less we're there, but I've not drawn something quite key on here. So why do we do chemiosmosis? Well, it's to power the joining of ADP and inorganic phosphate to make ATP. So this is the purpose, and we make loads of ATP here. I forget the 1s and 2s and 3s and 4s that we make during the substrate level phosphorylation. We're making lots. The numbers are a little bit variable, so I won't go into them here, but you make lots of ATP. We have a slight issue though, because now we've got, in order for this to continue to keep pumping protons here, we need a continuous flow of electrons, and we're going to get a whole load of electrons here. And if this were to continue, then we'd end up with lots of hydrogen here. We'd reach equilibrium. There'd no longer be a gradient. It would stop. So we've got to remove these products. How do we remove those products in order for it to continue? Well, there's an easy answer to that. In fact, let's put some numbers in here. There's 2 hydrogens, 2 electrons, and we're going to make. So these are our products. We've got ATP and H2O, and the most important thing we can say here is that oxygen is the final electron acceptor. This is going to pick you up marks all the time, this little key phrase here, this sequence of key terms. Okay, so let's put this into some note form. So we're going to say, starting here, that electrons and hydrogen or protons are released from coenzymes. So let's just put that down because people struggle with these coenzymes all the time. So this is the reduced form. They've got the hydrogen and then they split into their component parts, which we're going to call electrons. We're going to have hydrogen ions or protons. Again, you can use those two terms absolutely interchangeably. And then these are obviously regenerated. They're not going to be wasted or broken down. They're going to go back into the earlier stages of aerobic respiration to be reused. We can say, next stage, that the electrons pass along the electron transport chain in a series of redox reactions. So this releases energy, which is used to pump protons into the intermembrane space. Or maybe we should just actually say that it is used to create a proton gradient. What next? Well, the protons diffuse through ATP synthase in chemiosmosis. So this process is called chemiosmosis. And it provides the energy to combine ADP and PI to form ATP. And then finally, we need to mention the oxygen that allows it all to continue. It is oxidative phosphorylation. Oxygen is the final electron acceptor. It combines with electrons and protons to form water. So that is it. This is how we make nearly all of our ATP, oxidative phosphorylation. Some textbooks, old textbooks, will call this whole process electron transport chain. As you'll probably understand by now, electron transport chain actually refers to an electron flowing through electron carriers in a membrane. It takes place in photosynthesis and respiration and some other places. So the correct name for this stage of aerobic respiration is oxidative phosphorylation.
To get more biology videos from me, subscribe to the channel, and of course don't forget to try out my complete A-level course in the free trial over on the Tailored Tutors website. Up next, you can transform your grade in about an hour with my grade transformations playlist, or you can watch the video that's been magically selected for you from the people at YouTube.